Welcome you all in the new series of lectures on protein metabolism. This is the amino acid chemistry, and here we are going to discuss structure, classification, and significance. So, let's start with the structure of amino acid. So, what are proteins? Proteins are the linear polymer of amino acids which are linked together by peptide bond. So, the amino acids are the building block of proteins, and here in a biological system can produce protein with different properties and functions. For example, we have albumin, globulin, we have a fibrinogen, insulin, all the proteins with the different properties and different function. So these proteins, it has a function which is depend upon the type of gene which is present, and the type of gene which by the process of transcription and translation what it produce, and the produced protein which has a different functions. So what are the amino acids? So this is the general structure of amino acid, where you can see the carbon atom. This carbon atom is called as a chiral carbon atom, to which two groups are attached, that is amino group and carboxyl group, and hydrogen atom and a side chain. This carbon atom which is attached to four different groups, that's why it is called as a chiral carbon atom. And here you can see this is the R chain. This R chain, this is different for different amino acid. The amino acids differ from each other in their side chains. So on the basis of this side chain, we have a 20 different amino acids. So here what is the peptide bond? The peptide bond which is formed between the two amino acid, that is a carboxyl group of one amino acid and amino group of another amino acid. So if you want to a peptide bond, for that we need two amino acid having carboxyl group and amino group. Now here you can see that whenever the pH is 7.4, at this pH there is a typical Zwitter ion form of amino acid can be seen. The pH at which protein molecule exists as a Zwitter ion or dipolar ion or isoelectric ion is called as a isoelectric pH. So at this isoelectric pH, protein possess equal positive and negative charges, and they are electrically neutral. Protein does not migrate in an electric field. Whenever this protein which is subjected to electrophoresis, the pH at which the protein carries for both positive charge and negative charge. Such protein it is called as a Zwitter ion, and here you can see the two forms, protonated form and a deprotonated form. Protonated form means it has gained H plus ion, and a deprotonated form means here it loses H plus ion. So here whenever we apply electrophoresis, this ion it will not move, and it will help in electrophoresis whenever we want separation of protein, and at this pH the protein will not move. Similarly, at low pH, that is here the protein or amino acid it will act as a base. It has tendency to gain H plus ion, and here it act as a cation. So at a low pH the protein will carry positive charge. Similarly, whenever the pH on a higher side, here you can see that the protein or amino acid can behave as an acid. It has tendency to lose H plus ion, and here it carries negative charge. It has a very important significance whenever the patient is suffering from acidosis and alkalosis. In acidosis, the protein which is present in your body, that is a globular protein or a moving protein like albumin, at low pH the protein or albumin it carries positive charge, and at a alkaline pH, that is in metabolic alkalosis, the protein will carry negative charge. Now here you can see that the general structure of amino acid, where you can find the L amino acid and D amino acid. Depending on the orientation of amino group on alpha carbon atom, two stereoisomers are there. It is L and D isomers of each alpha amino acid exist. An amino acid is said to be L isomer if the amino group it is present on the left hand side. If it is in left side, it is called as the L alpha amino acid. Whenever the amino group is present on right side, it is the D amino acid. Here you can see that our body contains L amino acid. Generally, amino acid present in a protein are L form. That's why these are the L amino acid, which is commonly seen in a mammalian tissue. Sometimes what happens is a D-serine and D-glutamate, which is present in a brain. And D-serine and D-glutamate, these are the D form. Now here you can see that the numbering of amino acids. Here the carbon atom which is to which the carboxyl group is attached, that carbon is called as a alpha carbon, and next carbon is a beta, then next one is a gamma, next is a delta and epsilon. This is the structure of lysine where you can find these different types of carbon atom. Here we have a 20 amino acids. So here the 21st one is a selenocysteine and 22nd one is a pyrrolysine. Now here the classification of amino acid. First one is based on chemical structure of amino acids. Second one is a classification which is based on the nutrition, that is a nutritional classification. Third one is based on polarity of the side chains. And fourth one is based on the metabolic end product of amino acid metabolism. Now the classification which is based on the chemical structure of side chain. So here you can see that first one is the aliphatic amino acids, the classification in which we have a hydrocarbons. Second one is a classification which is based on the hydroxyl amino acid. Third one is a amino acid which contains sulfur, called as a sulfur containing amino acid. Fourth one is the acidic amino acids and their amides. Fifth one is the basic amino acids. Sixth one is the aromatic amino acid, means these are the benzene ring containing amino acids. And seventh one is the amino acid. It means that contains amino group instead of amino group. Now here in this series, first one is a aliphatic amino acid, that is a glycine. Glycine, it is a optically inactive, and here the carbon atom, that is the central carbon atom which is attached to same hydrogen. It has a two hydrogen on either side. It is smallest and simplest amino acid, and here you can find a three-letter code of glycine, that is a GLY. Here you can see the three-letter code is a GLY and one letter code is a G. Next one is a aliphatic amino acid, which is unbranched amino acid, and this is a alanine. Alanine, it has a three-letter code is ALA and one letter code is A. And here you can see the carbon atom which is attached to four different group, and it is optically active. The difference between glycine and alanine is on the basis of side chain, where in glycine we have a hydrogen and here we have a methyl group which is attached on a side chain. Now next one is a aliphatic amino acid. This aliphatic amino acid, it is a branched amino acid, and here we have a three-letter code for valine is a VAL or simply V. Here you can see this central carbon atom which is attached to three another carbon atom, and here you can see it is a branch. This is a branch structure, that's why it is called as a branched amino acid. Now here you can see this is a fourth aliphatic amino acid. This is a branch amino acid and it is a leucine. It has three letter code LU and simple L letter, that is a one letter code. Now here you can see a branching is here, and here we have a four carbon atoms are attached. Next one is a aliphatic amino acid. Again it is a branch chain amino acid, and here it is a isoleucine. It is the three letter code is ILE and one letter code is L. Now here you can see the methyl group which is attached to beta carbon in beta carbon, and here this is called as a isoleucine, as we have seen in the leucine where it is attached to a gamma carbon atom, while here the methyl group which is attached to gamma carbon atom, while here you can see that the methyl group which is attached to beta carbon atom, that's why it is called as a isoleucine. Now here you can see the glycine, alanine, valine, leucine and isoleucine, and it is GAVLI. This is a GAVLI. This is a mnemonic for the aliphatic amino acid, where you can see the G which is optically active, and there is a no chiral carbon atom here, while alanine, valine, leucine, isoleucine, these are optically active. And here you can see valine, leucine, and isoleucine. These are the branch chain amino acid. These are the branch chain amino acids. Then we have a hydroxyl amino acid. The amino acid in a side chain having the OH group. So in this series, here we have a serine and threonine. Here we have a two amino acid, that is serine and threonine. The three letter code for serine is SER and one letter code is S. Similarly for threonine, the THR, that is three letter code, and one letter code is T. So here you can see the OH group is attached in serine and threonine. These are the hydroxyl amino acid or side chain having OH group. Then we have another amino acid in this group, that is a hydroxyl amino acid. This is a tyrosine. Actually the tyrosine, it is a aromatic amino acid, but it has a phenolic OH group which is attached at a para position of this benzene. This, this is a para position here, and it is a amino acid which has OH group. Then now here you can see that this is a sulfur containing amino acid, methionine and cysteine. Methionine, it has a three letter code is MET, and M, it is a one letter code, and here you can see that we have a sulfur atom, and here there is a no hydrogen atom. Here hydrogen atom, it is replaced by methyl group, and here this is a methionine structure, while in case of cysteine we have a sulfhydryl group is here. In case of methionine, there is no sulfhydryl group, only the sulfur group is there, and here there is a sulfhydryl group, it is present in a cysteine. Then we have a cysteine, it is also a sulfur containing amino acid, where it is a combination of two molecules of cysteine. Whenever two molecules of cysteine get condensed, we get a cysteine. Now we have a acidic amino acids and their amides. So we have aspartate and glutamate. And here you can see that we have a carboxyl group which is attached to beta carbon atom. So it is called as a beta carboxylic acid. Aspartate, it is called as a beta carboxylic acid containing amino acid. This is the aspartate. And here this is a the carboxyl group which is attached to gamma carbon atom. So here in this aspartate and glutamate, here we have a 2 carboxylic acid group. That's why it is called as a dicarboxylic monoamino acid. You can see here we have a dicarboxylic acid. This is the dicarboxylic acid and monoamino group. You can see a monoamino group. That's why it is a dicarboxylic monoamino acid. Next one is the amide of this aspartate and glutamate is aspargine and glutamine respectively. In case of aspartate, we have a COOH group, and here OH group is replaced by amino group. These are the amide of aspartate, and here glutamine it is the amide of glutamate. And here the code is ASN for aspargine, and N it is a single letter code, and GLN for glutamine, and single letter code is the Q. So aspartate, glutamate, aspargine and glutamate, all 4 amino acids are different, and the codon which codes for these 3 different amino acids, it's 4 different amino acids, is different. They are not same as for aspartate and aspargine and for glutamate and glutamine. Then we have a basic amino acid. This is the histidine. So histidine is a R chain or side chain. It contains a imidazole ring. The imidazole group is attached to the side chain in the case of histidine, and the 3 letter code is HIS and 1 letter code is H. Similarly, in lysine, we have a LIS. The 3 letter code is LIS and 1 letter code is K. And here you can see that the amino group is attached on the epsilon carbon. That's why it is called as the epsilon amino group. Lysine, it is a epsilon amino group containing, it has a amino group at a epsilon carbon atom. Then the basic amino acid is also arginine. Here you can see in arginine we have a additional side chain, and side chain which contains guanidino group. Here you can see this is a guanidino group. This is a guanidino group which is a specialty of this arginine. Then we have a aromatic amino acid, that is a tyrosine, phenylalanine and tryptophan. The first one is tyrosine, which we have already discussed in a basic amino acid group. Here we have a phenolic OH group of tyrosine is here. So this tyrosine, it is a non-essential amino acid, and the starting material for tyrosine is a phenylalanine. So in phenylalanine there is a no OH group, and here you can see this is the OH group. So in case of phenylalanine there is a no OH group, and in case of tyrosine there is the OH group. This is the difference between tyrosine and phenylalanine. Now here you can see that in case of phenylalanine there is no OH group. There is a no OH group as like a tyrosine. Here there is a no hydroxyl group, and it contains benzene ring. That's why it is an aromatic amino acid. And here you can see that the phenylalanine, it is a essential amino acid, while tyrosine it is a non-essential amino acid. So phenylalanine get converted into tyrosine by the action of enzyme phenylalanine hydroxylase. Next one is a tryptophan. Tryptophan, it has a additional side chain, contains indole group. Here you can see this is a indole group. It is present in aromatic amino acid. The specialty of this tryptophan is, here you can see there is a indole group. Then the last amino acid in this series, it is a proline. It is the 3 letter code is pro, and 1 letter code is a P. And here you can see it has a imino group. Here there is not a amino group. This is a imino group is here, and it has a pyrrolidine ring. This ring is called as a pyrrolidine ring, and this is a amino acid, not amino acid. So these are the three-letter code and one-letter code for all 20 amino acids. Okay. Now we have a 21st amino acid, that is a selenocysteine, and the three-letter code is SEC and one-letter code is U. So here the structure is same as that of cysteine. In cysteine we have a SH group. We have a SH group is here, sulfhydrylase group is there. But in case of selenocysteine, the sulfur group it is replaced by selenium. That's why it is called as a selenocysteine. And this selenocysteine it is coded by UGA codon. Actually, the UGA it is a stop codon, but sometimes this UGA it can code for amino acid, that is a selenocysteine. So here you can see it has selenocysteine. It has a participate in activity of this enzyme. The enzymes are glutathione peroxidase, deiodinase, glycine reductase, and thioredoxine reductase. So this may be a SAQ in your MHS exam. What are the four enzymes for the activity of four enzymes? The cellular system is required. Cellular system participate in activity of glutathione peroxidase, deiodinase, glycine reductase, and thioredoxine reductase. Now here, this is a 22nd amino acid, that is a pyrrolysine. It is a stop codon UAG that can code for this 22nd amino acid pyrrolysine. Now in this we have discussed about all 20 amino acid with additional 21st and 22nd amino acid. So this can be a MCQ, the stop codon UAG codes for which amino acid, so your answer will be pyrrolysine. Now, now the classification based on the nutritional requirement. So the essential or indispensable amino acids are the amino acid which cannot be synthesized by the body. It means that there are some amino acids which are not synthesized by the body, and such amino acids are need to be supplied through diet. Such amino acids are called as essential amino acids. There are some conditions like in a growing age, during pregnancy, during lactation, and during recovery from the prolonged illnesses. So as in case of growing phase, as a phase here there is a proper growth and development in this growing phase, which is very important. So for this activity we need all essential amino acids. Similarly, during pregnancy the fetus is growing inside the uterus, and for proper intrauterine growth and development we need essential amino acids. That's why the demand of amino acid is increased during pregnancy. And during pregnancy, if you want your baby healthy, the pregnant lady should consume essential amino acids. Similarly, in lactation, again the condition is same, as the fetus is total as the baby it is totally dependent on milk for nutrition. So during lactation, it is very important that mother should take healthy food. With the healthy food in the sense the food which is rich in a protein, carbohydrate. So the protein should be rich in essential amino acids, so that the essential amino acid that will go to baby for proper growth and development. Similarly, recovery from prolonged illnesses. What is the meaning of recovery from prolonged illnesses? If any patient who is suffering from tuberculosis or from cancer therapy, or by that is a chemotherapy, or who is suffering from cancer or any other major illnesses, or during the surgery, there is a big protein, big blood loss, and also there is a the surgery there was a there was a complication in surgery. That's why the patient stays in the hospital for a long period of time. During that condition, during recovery, we need a essential amino acids. So during this condition, we supply nutrition in a form of supplement, either orally or by intravenous route. Sometimes the patient who is not able to take food in that condition, it is necessary to supply essential amino acid by intravenous route. Here you can see that the 10 essential amino acid in an ascending order of dietary requirement. That is arginine, tryptophan, histidine, methionine, threonine, isoleucine, valine, phenylalanine, lysine, and leucine. The mnemonic for this essential amino acid is AV-HIL MPTT. Now here you can see that essential amino acid, the same list is here, but here we have a arginine and histidine. Where arginine and histidine, these are the semi-essential amino acids. So all other are essential amino acid, while arginine and histidine, these are the semi-essential amino acids. So why it is called as a semi-essential amino acid? Now here you can see that the non-essential amino acids are glycine, alanine, serine, cysteine, aspartate, asparagine, glutamate, glutamine, tyrosine, and proline. So this amino acid, it can be synthesized by the body from the essential amino acid and need not to be consumed through diet. So why the arginine and histidine is called as a semi-essential amino acids? So arginine and histidine can be synthesized by adult but not by growing children. Hence these are considered as a semi-essential amino acid. So the next classification is classification of amino acids based on the polarity. So here on the basis of charge and polarity of their side chains, the 20 standard amino acids have been divided into two main groups, hydrophilic and hydrophobic. Hydrophilic means these are the water lover and hydrophobic means these are the water haters. So in hydrophilic we have polar one, and in this polar we have a three group: negatively charged amino acid, positively charged amino acid, and uncharged amino acid. Now here you can see that this is a negatively charged amino acid, in which we have a aspartic acid, glutamic acid. In case of positively charged amino acid, we have a lysine, arginine, and histidine. While in case of uncharged amino acid, we have serine, threonine, asparagine, glutamine, cysteine, and tyrosine. So all these are hydrophilic amino acids. While in hydrophobic group, that is non-polar group, here we have aliphatic amino acids and aromatic amino acids. In aliphatic, we have glycine, alanine, valine, leucine, and isoleucine. While in case of aromatic amino acid, we have phenylalanine and tryptophan, and we also have proline and methionine. So let's discuss one by one. So now here in this group, in a hydrophilic group, first of all we will discuss negatively charged amino acids. So here we have a aspartic acid and a glutamic acid. This group include these two important acids which are acidic one, and here because of COOH group, that is a carboxyl group in this side chain. So aspartic acid and a glutamic acid that donate proton at a neutral pH, and it becomes negatively charged. Now here in this group we have hydrophilic amino acid which are positively charged, which includes lysine, arginine, and histidine. These are the basic amino acids. Because of amino group in their side chain, glycine and arginine accept proton at a neutral pH and it become positively charged. The side chain of histidine can either positively charged or uncharged at a neutral pH so that they can form a hydrogen bond. That's why these are the hydrophilic amino acid or polar amino acids. Now, here in this group, we have six amino acids which are uncharged, that is serine, threonine, asparagine, glutamine, cysteine, and tyrosine. These are all hydrophilic amino acids and these are the uncharged amino acids. Now, here you can see that the uncharged that is serine and threonine. The polarity of serine and threonine is contributed by hydroxyl group of that side chain. So here this hydroxyl group which participate in formation of hydrogen bond. That's why these are the hydrophilic or polar amino acids. Now in this group here our asparagine and glutamine, this contains amide group and this amide group which participate in formation of hydrogen bonding. That's why these are the hydrophilic amino acids. So here in this group we have a cysteine and tyrosine. This cysteine, whenever the two molecule of cysteine with a sulfhydryl group can covalently join together and they form disulfide linkage to yield cysteine. The disulfide linkage is formed by the oxidation of sulfhydryl group of two cysteine molecule and protein contains both cysteine and cysteine in their polypeptide chain. So both cysteine and cysteine determines the structural and functional properties of various proteins. So the cysteine it is a amino acid with a sulfhydryl group and it is a polar one and it participate in formation of uh, this hydrogen bonding. Similarly, we have a tyrosine. Tyrosine it has a OH group and this OH group also participates in a hydrogen bonding. Then we have a hydrophobic. These are the aliphatic amino acid, glycine, alanine, valine, leucine, and isoleucine. These are the aliphatic amino acids, and these are the hydrophobic because the side chain it contains hydrogen and carbon, and they never participate in hydrogen bond formation, or they are not a water lover. So there is a hydrophobic interaction. Similarly, in aromatic amino acid, the same thing is there. This benzene ring and indole group of tryptophan, they never participate in formation of bond with the water. Again, we have proline and methionine. The same thing is here. There is no free hydrogen atom here, and they never participate in formation of bond. So these are the hydrophobic amino acid. If the amino acid proline is there, it is responsible for making the alpha helix unstable because of this hydrophobic interaction. Now, the last classification is classification based on the metabolic product of amino acids. So, in this, we have ketogenic amino acid. So, what is the meaning of ketogenic amino acid? The amino acid, the end product of this amino acid after metabolism, they give rise to ketone bodies. So, the catabolic end product of this few amino acids are either acetoacetate or one of the precursor. So the precursor it may be acetyl-CoA or acetoacetyl-CoA. So acetoacetate is one of the three ketone bodies and the catabolic end products acetyl-CoA and acetoacetyl-CoA either convert into the ketone bodies. So the detailed expression we will discuss in a lipid metabolism where the steps involved in the ketone bodies formation. So the precursor for this ketone bodies is a acetyl-CoA and this acetyl-CoA is required is produced at the end product and acetoacetate is also produced as the end product of this leucine and lysine metabolism. So these are the ketogenic amino acids which give rise to ketone bodies. So what is the meaning of both ketogenic and glucogenic? So here in this group we have isoleucine, phenylalanine, tyrosine and tryptophan. The end product of this protein, these amino acids, these are the both glucogenic and ketogenic precursors. So these the end products are pyruvate or TCA cycle intermediate or acetyl-CoA and acetoacetate. Since this amino acid can be metabolized in both glucose and ketone bodies and they are termed as a glucogenic and a ketogenic. And the last one is whatever the remaining amino acids are the glucogenic amino acids and that give rise to glucose by the process of gluconeogenesis. So what is the meaning of gluconeogenesis? The formation of new glucose from the non-carbohydrate precursors. So here the catabolic end product of the remaining amino acids means out of 20 we have a 14 amino acids and that amino acids are glycine, alanine, methionine, threonine, arginine, glutamate, glutamine, aspartate, aspargine, cysteine, proline, serine, valine, and histidine. So the catabolic end product of these amino acids can be converted into glucose by the process of gluconeogenesis. That's why these are the glucogenic amino acids. So this is all about the metabolic products of amino acid, how they participate in the formation of ketone bodies and formation of glucose. So this is the end of this part. So I hope you like this lecture. Please like, share and subscribe. Thank you.